Hi to everyone, my name is Piera Della Porta, and my thesis work regards the production and characterization of MPEG PLGA nanoparticles for bedaquiline release to fight the multi-drug resistance. The experimental part of this thesis work has been performed at the Department of Pharmaceutical Sciences of Utrecht University during the Erasmus project. First of all, I want to introduce the problem of multi-drug resistance. That's a resistance shown by some microorganisms that have evolved a series of mechanisms to overcome the effectiveness of drugs, thus surviving exposure to the drugs. This implies high mortality rates, high medical costs, threats to therapeutics, but especially a decreased effectiveness of the drugs. The resistant microorganisms are, for the most, bacteria, and the most spreads are Staphylococcus aureus and Mycobacterium tuberculosis. My work is focused on the resistance shown by the second one. Tuberculosis is an infective disease spreads in all the world and it is cause of million deaths annually. The World Health Organization has estimated that a high percentage of treated and new cases show the resistance to the drugs normally used. Tuberculosis spreads via aerosols and typically attacks the lungs, but can also affect other body organs. The bacterium reaches the alveolar spaces and it is phagocytated from the macrophages that identify the bacterium as foreign and attempt to eliminate it. The bacterium can survive in the macrophages and also reproduce itself. The macrophage at this point is surrounded by other macrophages that form a solid structure called granuloma. The bacterium can stay latently in the structure without showing symptoms, or the structure can break and spread the bacterium in the rest of the body, especially in immunocompromised patients. The bacteria that cause tuberculosis can develop resistance to the two most efficacious antimicrobial drugs used to cure the disease. For this reason, new antibiotics were introduced. Bedaquiline, that belongs to the class of the arylquinolin, has shown to be efficacious to fight also the resistant bacteria. In relation to that, it has to be introduced a window of selection mutant existing between two concentrations, the minimum inhibitory concentration and the mutant prevention concentration. To be sure to kill all the population of bacteria, including the mutant ones, it's necessary to reach, at the infective site, a concentration that is higher than the mutant prevention concentration. This can be not easy using just the free drug because of the poor distribution and the fast elimination. So, in order to keep an high systemic concentration and a concentration higher than the mutant prevent concentration in the infection site, delivery systems were implemented, and in particular nanoparticles that are capable to reach the air cells. So, the aims of my thesis are the preparation and characterization of nanoparticles loaded with bedaquiline, prepared varying the drug to polymerization and with different concentration of surfactant and with different preparation methods, evaluate the release in the different cases, and try to introduce a new approach for the release study. I have already introduced the multi-drug resistance and the aims of my study. Now I want to set about explaining the material and methods used and discussing the results. The copolymer that has been used for the nanoparticles formulation is formed by two blocks, one hydrophilic, represented by the MPEG, and one hydrophobic, represented by the PLGA. The PEG covers the external part of the nanoparticle, that's a graft ligand, and permits the lung circulation, while the PLGA, contained internally, interacts with the hydrophobic drug. The copolymer is biocompatible and the degradation products are easily eliminated by normal metabolic pathway. The used drug, bedaquiline, is a new compound with a new mechanism of action. In fact, it's able to inhibit the C subunit of ATP synthase enzyme of the bacterium that is really important for the energy production. So the drug can work when the bacterium is active, but also when it is latent, because anyway, it needs, and in any case, it needs energy. The compound is hydrophobic, as is shown by the repartition coefficient, and it has a good binding with the plasma proteins. The preparation method for the nanoparticles is the single emulsion solvent evaporation. So there is an aqueous phase containing a surfactant, polyvinyl alcohol, at which it is added an organic phase of dichloromethane in which polymer and antibiotic were previously dissolved. After a sonication phase and the phase of evaporation of the organic solvent, the solution of nanoparticle was obtained and subjected to a washing step and centrifugation to remove the possible residue of surfactant from the nanoparticle surface. This is a standard method, and it is from the study release. It was noted the presence of a fast release at the beginning. That's why other preparation methods to limit the fast initial release were carried out. In particular, on the base of the standard method, some compounds were added to try to improve the bond between the nanoparticles and the hydrophobic drug, and also to remove the possible drug present on the surface of the nanoparticles. And furthermore, a nanoprecipitation method was used. The nanoparticles were characterized in terms of average size, zeta potential, and morphology. The loading capacity and the encapsulation efficiency were determined using UV and HPLC analysis. The release study was carried out using two different dissolution complex media. Middlebrook 7H9, that's normally used for the tuberculosis mycobacterium culture, and it is poor of proteins and lipid. The RPMI, used normally for the macrophages culture, was enriched instead with proteins through the adding of fetal bovine serum. This a complex medium. This one is really similar to the physiological condition. The nanoparticle solution was added to the complex media and kept in agitation at 37 degrees. At appropriate intervals of time, an aliquot was collected after centrifugation, and new fresh media was added. The taken supernatant was stored at minus 20 degrees to be subsequently analyzed for the release evaluation. In addition, a new approach to the release study was introduced because until now the release is always evaluated in the bulk of the dissolution medium without taking into account the drug residue present in the carrier. So, first of all, a separation between the carrier and the complex medium was carried out using a site exclusion chromatography ACTA purifier. And consequently, analytic methods were used to determine the amount of proteins in the two separating phases. And in order to verify the separation finally, a spectrophotometric analysis was carried out to analyze the eluids in exit from the chromatographic column. So, first of all, the copolymer was characterized by an NMR analysis. The resulting peaks correspond to the molecular composition of the copolymer, and it was possible to calculate the molecular weight and compare it with the theoretical one. And more, the percentage of PEG and the ratio lactic acid and glycolic acid were calculated. The nanoparticles obtained for different feed ratio were characterized in terms of sites, showing sites included in the desired range. The value increased slightly by increasing the amount of drug. The zeta potential obtained show that the nanoparticles are neutral, and the encapsulation efficiency is quite high, except for the last case where the amount of drug is the highest one. For the ratio 1 to 10, the morphology was analyzed using the transmission electron microscopy. The nanoparticles show a spherical shape, and the measured sites correspond to the one obtained by the DLS analysis. On the base of the standard method, on the standard method preparation, the last washing phases were changed to try to remove the eventual drag present on the surface of the nanoparticles. So, first of all, the solubility of the drug was investigated. The drug is insoluble in water and in most of the organic solvent, if not slightly in methanol. So different solutions of methanol were used in the washing step, and the nanoparticles obtained were characterized, showing more or less the same results for the dimension and encapsulation efficiency, except for high percentage of methanol, where the encapsulation efficiency decreased. So, because of obtaining the same result, it was preferred to carry on using water for the washing steps, also to not interfere with the polymeric structure of the nanoparticles. Furthermore, the nanoparticles were prepared varying the concentration of surfactant, PVA, using higher concentration compared to the normal used one. It's clear that the results for the encapsulation efficiency are the same for both the concentration, while the size results are slightly different. In fact, the size for the 5% concentration is smaller than the size for 1% concentration of PVA because of the stability of the nanoparticles increase. More, the nanoparticles obtained with different preparation methods were characterized. The value in terms of size and encapsulation efficiency were acceptable and similar to the ones obtained for the standard method, except for the case of the nanoprecipitation, in which the size is included in the micrometric range and not nanometric anymore. After that, the release study was carried out, and in this slide the release results in Middlebrook 7H9 are shown. This medium is the less proteic complex medium, and the results for the different concentration of PVA are compared. And despite the size diversity, there is no change in the trend for the two cases. That's because of the difference in size is really small. The total release is not achieved, but only the 60% of the total, and there is a fast release already in the first 2 days, where the 50% of total is achieved. Regarding to the other complex medium, more proteic and closer to the physiological condition, the total release is achieved. And also in this case, it's clear that the trend is the same for the two cases of concentration of PVA. In this case, the initial release is fast as well. The predominant release mechanism is the diffusion and the surface runoff. The erosion mechanism is not evident, also because monitoring the sites during the release period, it's clear that there is not a decrease in the nanoparticle size, except maybe in the last period of the release, around 18 and 20 days. Using the same release methods, the release for the nanoparticles prepared with different techniques was carried out. And in this case, the purpose was the attempt to reduce the fast initial release using some variation in standard method. But comparing the results obtained with the standard one, it's possible to notice that there is no distinction between the two cases. The value of cumulative release is the same after 2 days, and the trend is the same as also the predominant mechanism. Furthermore, the same behavior influenced by the dissolution medium is evident. In fact, also in this case, with the less proteic medium, the total release is not achieved. Instead, for the more proteic medium, where the presence of proteins changed the release pattern, we can see that the total cumulative release is achieved. And so, in summary, it appears no evident side effect. The predominant release mechanism is the diffusion, but an initial fast release is evident in every medium and in each study cases. Surely there is an important effect of the dissolution medium. In particular, the presence of protein helps to maintain always high the concentration gradient, thanks to the bound of protein to the drug, that is in fact always subtracted from the equilibrium. Afterwards, it was evaluated the new approach to study the release regardless of the dissolution medium. Until now, the release study is always being focused on the analysis of drug present in a dissolution medium. And in order to obtain a total balance on the drug amount, it was tried to evaluate also the residual amount of drug in the nanoparticles. For this reason, a solution of the only carrier systems was injected in the chosen analytical instrument, the ACTA purifier, and as it showed, two different carrier systems were injected separately, a solution of liposomes and a solution of the used nanoparticles. Thanks to the instrument, it was possible to obtain a characteristic peak for both the carriers, and at this point, a solution of just the dissolution medium was injected. In this case, FBS was chosen. The fetal bovine serum is a medium rich of proteins. And finally, a solution of the carrier dispersed in the medium was injected, and it's already evident for both the carrier system that there is a separation between the peaks relative respectively to the delivery system, MPS or liposomes, and the complex medium. Afterward, the injected solution of carrier in complex medium was collected, ml by ml, and analyzed by a colorimetric spectrophotometric method. Thanks to that, it was determined the amount of proteins in the two fractions. The reaction with the reactive doesn't happen in the MPS fraction, while it is visible that the change of color in the wells belonging to the dissolution medium fraction as an acknowledgement that there are not proteins in the nanoparticle fraction, but only in the medium. And it confirmed the occurred separation, as also the overlapping of the chromatograph by ACTA with the evaluated concentration in the fraction by the method shown. Once evaluated a separation, it was attempted to determine the amount of drug released in the medium and in the residual one in the carrier. The injected sample was eluted and collected in the two fractions, and after using two different methods of extraction for the drug, it was analyzed by HPLC. The recovery percentage respect to the theoretical initial value of the drug in the solution is higher in the nanoparticles fraction, but it exhibits very low values, revealing a loss of drug. In order to verify the complete exit of the eluids from the column, two model compounds conjugated with fluorescent substance were utilized, respectively one for the nanoparticles and one for the medium, and once injecting in the column and collecting the solution were analyzed by a spectrophotometer to determine the percentage of recovery respect to the initial theoretical value of injection. The results indicate that the model compound for the solution medium was completely collected at the exit of the column, while the one related to the nanoparticle system was partially recovered. So this implies that part of it remains in the column. In conclusion, the polymeric nanoparticles were formulated and prepared successfully, showing a high encapsulation efficiency with sites in the desired range. The use of methanol doesn't help for the cleaning of a nanoparticle surface, showing the same results of the standard washing water method. The surfactant concentration doesn't influence the drug release, and that that is strictly influenced by the presence of proteins in the dissolution medium. Also that the predominant mechanism for the release is the diffusion, showing an initial burst release that despite the use of other preparation techniques for the nanoparticles, it is not possible to eliminate. It was obtained a separation between the complex media and the nanoparticle system, verified by the no presence of proteins in the nanoparticle fractions. For the future, further optimization of the collecting methods should be done, and another analysis method of the drug in the fraction should be evolved. Thanks to all for the attention.